First, I'd like to start by thanking the organizers for the invitation to speak today. As an early stage investigator, this is an excellent opportunity for me to present my work to such a fantastic group of myeloid biologists, and so thank you for the opportunity. Today I'm going to be discussing the work that I have been doing on dynamic dendritic cell-mediated antigen packaging and dispersal. This work that I'm going to present is kind of divided into two parts. First, I will go through some of the previously published work that I published about a year ago as I was exiting my postdoc at UCSF with Max Krummel, looking at melanoma-derived tumor antigen dissemination in the lymph node and the impact that that had on anti-tumor T-cell priming characteristics. With that, I'll go into a little bit of detail on some of the tumor models that we use to track antigen and that I'm still using in my independent group to focus on the fate of antigen in different dendritic cell populations. So in part two, I'm going to share, and I'm excited to share some new data that's unpublished that is serving as kind of the foundation of my new independent group at Oregon Health and Science University, trying to hone in on the impact of source and context of antigen acquisition on the function of dendritic cells and antigen presentation. So I always like to start off with just touching on and reminding everyone of the importance of the lymph node in generating anti-tumor immunity. So in the tumor itself, there are critical changes that occur that are essential for the detection of the tumor by the immune system, one of those being the misexpression of self-proteins or the expression of neoantigens that can be taken up by professional antigen-presenting cells and the TME and drained into the tumor-draining lymph node. And then these professional antigen-presenting cells will process that antigen and present it on MHC molecules, where you get your very first interaction with a cognate T-cell receptor. These naive T-cells are then primed. They can expand, leave the lymph node, go back into the tumor microenvironment, and execute their cytotoxic function. So while, of course, the immune response in the periphery and the cytotoxic T cells in the periphery are really the business end of the immune response, in order to understand the type of T cell repertoire that we have at our disposal for mediating anti-tumor immunity, we need to understand in great detail the consequences of differential priming events in the lymph node. So the identity of the dendritic cells that drive the priming and the functional and dysfunctional properties of that priming event. So for many years now, the Krummel lab at UCSF has been interested in understanding how antigen arrives in the lymph node and how different dendritic cell populations result in productive anti-tumor immunity. I'm specifically looking at conventional dendritic cells. So these conventional DCs can be divided into CDC1 and CDC2 subsets, where in a simplistic view, you can think of CDC1s as driving CD8 positive T cell responses. There are specialized and cross-presenting antigen on MHC1 molecules, whereas CDC2s are thought of as the canonical drivers of CD4 T cell responses. When we go into the lymph node, things get more complicated in that CDC1s and CDC2s have migratory and resident subsets, where migratory CDC1s are marked by CD103 and migratory CDC2s by CD11B. The migratory populations are those out in the peripheral tissues that can acquire antigen in tumor and in tissues and bring that antigen into the lymph node. Whereas resident DC subsets are considered local permanent residents of the tissue lymph node microenvironment. So they don't acquire antigen in the periphery. Instead, any antigen brought to them, any antigen that they have is brought to them by cell-mediated trafficking or by drainage in the lymphatics. These CDCs, in the case of CDC1s, are marked by CD8 alpha, and in the case of CDC2s, I'll refer to them as resident CD11Bs. So previous work by the Krummel group had shown that a myriad of myeloid populations can acquire antigen in the periphery, and that arrival of antigen in the lymph node is dependent on CCR7 mediated migration of these myeloid populations into the lymph node. So cell-mediated migration is required for the trafficking, and that then CD103s really drive your dominant CD8 positive T cell response. But some interesting findings that came from this work really centered around the distribution of antigen in tumor draining lymph nodes. What I mean by that is that when we were doing these studies using a fluorescent protein expressed within the tumor cells, you could find tumor-derived material being brought in by the migratory populations, but it wasn't simply restricted to migratory DCs. And instead, you could find fluorescent protein present in these resident DC populations. So resident CDC1s and resident CDC2s were positive for this green fluorescent protein, suggesting that antigen is shared with other cells once it arrives in the lymph node. So the questions then stemming from this were, how exactly is antigen moving from one cell to the other? And once inside of a resident DC, what is the impact for T cell priming? So do resident DCs have some type of functional impact, the same or different from their migratory counterparts? And so when I was doing my postdoc with the Chroma Lab, we designed some systems to try and answer these questions. So using B16 melanoma lines expressing a ZsGreen as well as the model antigen OVA, we could durably track antigen as it moved within different DC populations, and then also do T cell specific, TCR specific stimulation experiments. So allowing these tumors to grow and then excising the lymph nodes and doing flow cytometry to look for tumor-derived ZsGreen. We found that ZsGreen is indeed readily shared with resident populations in the lymph node. So here you can see this flow cytometry quantification where CD103 migratory DCs and migratory CD11B DCs are positive, both at around 40% of the population positive for ZsGreen. And this is not simply restricted to those cells. Instead, you find resident DC populations like resident CD11Bs and resident CD8 alpha DCs are also positive for this tumor-derived material, and at a proportion very similar to their migratory cells. So around 40% of these populations are also positive for ZsGreen, demonstrating that antigen is being readily shared. So again, the question is, how is it moving and what does it mean for T cell responses? And so we did a number of different imaging experiments, but to start off, I'll share with you the data from in vivo. So can we actually capture the event of antigen trafficking in vivo and visualize the passing process to get an idea of how that antigen is moving? So we used a triple reporter mouse that expresses XCR1 Venus, CD11C M cherry, and CSF1 receptor drives the expression of CFP. And using this triple reporter mouse, we give them B16 ZsGreen tumors and then look at the tumor draining lymph node and see if we can capture that passing event. Here I'm showing you two stills from the tumor experiments where you can see an XCR1 positive loaded CDC1 that will interact with a variety of different myeloid cells at these different two still time points. And then in the actual imaging experiment, we can capture that passing event. So here you have a M-cherry positive CDC2 loaded with ZsGreen interacting with an XCR1 positive CDC1, and you will see that event of antigen passing. So here's your CDC2 positive for M-cherry interacting with your CDC1, and the antigen essentially seems to split off and divide in between the two cell types. So here it plays again and it shares. So what was interesting about this is not only can you visualize the passing at a junction point, so it seems as if the sharing of antigen is dependent on a formation of contact between two DCs, but also that when the antigen is shared, some of the information is still retained, some of the antigen is still retained within what we call the donor DC or the originally loaded DC, suggesting that this process serves as a way to propagate antigen throughout the populations of the lymph node. So we wanted to look at the identity of the passed antigen in a more refined context. So getting an idea of how the context in which that antigen is packaged and passed from cell to cell. So using a membrane tomato expressing mouse, we injected B16 ZsGreen tumors, excised the tumor draining lymph node, and then could use lattice light sheet imaging to get high resolution detail of how the antigen is held on a single cell level. So these are migratory CD11b DCs that we've sorted from the tumor draining lymph node. Here I'm showing you just a Z slice where you can see the bits of ZsGreen intracellularly. And then here, this is a surfaced DC where we use a program called Chimera X that allows you to peel back layers of your signal. In this case, we're peeling back the surface membrane tomato signal until you hit the these little bits of ZsGreen. When you turn this to the on-phos view, you find that these bits of ZsGreen are encapsulated by membrane, showing a vesicular form of antigen. So within a migratory DC that has tumor-derived antigen, the antigen is encapsulated within membrane tomato positive cell membrane. And then we were curious as to whether or not this antigen packaging was then transferred as the tumor-derived antigen was passed from one cell to another. So is the passing actually through an intact unit of vesicularized antigen? So to get at that, we used, again, these membrane tomato mice with B16 ZsGreen tumors, but we sorted out the ZsGreen positive DCs. So essentially sorting out these cells here, and then co-cultured them with a congenitally marked, unlabeled recipient DC. So just a CD45.1 DC. Then incubated them for 16 hours in a co-culture. And then from that co-culture, sorted out CD45.1 positive cells that were now ZsGreen positive, and then did lattice light sheet imaging to look at how that antigen was being held in the recipient DC. And what we found was really exciting in the context of understanding how antigen is encapsulated and shared, in that when you stain these CD45.1 cells with a membrane, here the membrane is just stained with CD45 APC, so you have purple, pseudo-colored purple, and then here in a Z slice, you can find your bit of ZsGreen intracellularly. And when you look at it in this 3-color antigen sharing experiment, you find that that antigen retains that membrane tomato signal surrounding that bit of antigen. So the sharing of antigen is actually through the sharing of these vesicularized units. So this is just that same imaging surfaced and rotated, so you can see it even better. Here, that ZsGreen particle encapsulated in your membrane tomato from your donor host mouse, all within this new recipient DC. And so now that we have all this interesting data about how antigen was being shared in the physical space, we were seeking to understand what that meant for T-cell priming. So since the ZsGreen tumors contained a model antigen, we could sort the DCs based off of antigen bearing versus antigen non-bearing, so ZsGreen positive versus ZsGreen negative DC subsets, and then co-culture them with OT1s that had been labeled with a proliferation dye and look for differences in T-cell proliferation. So after 72 hours, our readout would be a dilution of our proliferation dye. So sorting based off of ZsGreen positivity gave some expected and some unexpected results. So in the case of sorting ZsGreen negative cells, these cells don't have antigen because they don't have ZsGreen, which is marking the ova. And you see that there is no stim, so similar to an unstimulated OT1. But when you sort out the ZsGreen positive DCs, you get what you expect in the case of CD103s, in that these are proficient primers of OT1 T cells. You get lots of proliferation in these co-cultures, but surprisingly, we saw very efficient stimulation of OT1s by both M11Bs and CD8 alpha resident CDC1s. So this suggests that these resident CD populations that are receiving antigen in this vesicular passing mechanism are actually proficient at priming T cell responses in the context of this in vitro T cell proliferation assay, and nearly to the same extent as a CD103. And so given that these resident DCs could have could either have equivalent downstream anti-tumor T cells, or it could have some type of subset specificity. We were curious at looking at the identity of these T cells following different priming by different DC subsets. And what we found, what we decided to do was look at gene expression profiling from the T cells following subset specific stimulation. And so looking at bulk RNA-seq of the cultures following the different types of stim, you found that while CD103s and migratory 11Bs, so these migratory DC populations stimulating T cells, had relatively equivalent there were some similarities with the T cell priming that were primed off of these DC subsets. When you look at CD8 alpha resident DCs, you saw a really distinct gene expression profile from the T cells. And the genes that were being differentially expressed have really interesting potential functional consequences. So the types of genes that were downregulated after a CD8 alpha stim compared to, say, a CD103 DC stim were things like response to interferon gamma, pathways for cytotoxicity and immunity, and pattern recognition receptor signaling. And so just to summarize the findings from this work, and as I mentioned, this is published, and so I didn't have time to go into the details, but you can certainly find it in the publication, this being the graphical abstract showing that we found antigen uptake and trafficking to the lymph node resulted in contact-mediated sharing of antigen in these vesicular units with resident DC populations, and that this antigen within the resident DCs resulted in a distinct T cell priming characteristics that were indicative of suboptimal priming. And so in my current lab, we're interested in looking at the characteristics of this suboptimal prime and whether or not it is impactful for anti-tumor immune responses out in the tumor microenvironment. But this work within the context of tumors also brought up a lot of questions that have kind of served as some of the groundwork of my own independent lab, looking at the impact of different sources of antigen on DC function. And so and we'll move into part 2, discussing source and context of tumor or of antigen acquisition. And with that, I'll introduce a couple of new model systems that I've been using. So we look at tumor-derived ZsGreen as a way to track tumor antigen, but I also have ways of tracking steady state self-antigen using ZsGreen as a surrogate as well. So by crossing ZsGreen mice with a K14 Cree, we can get skin restricted expression of ZsGreen, and we can then track self-antigen and the patterns in which that antigen disseminates in the lymph node. Here I'm just showing you from the tissue itself, so antigen uptake in the tumor and antigen uptake in the skin, and the patterns of antigen uptake are fairly similar. So though it's to a somewhat lesser extent, you still see, proficient loading of DCs, both M11Bs and CD103 DCs readily load with antigen even in the steady state skin, suggesting there's a constant sampling of self-antigen in a similar way that DCs take up tumor antigen. But what's interesting is that when you move to the tumor draining lymph node and the skin draining lymph node, you start seeing some pretty distinct differences in how antigen is available to DCs. So within the tumor, and this is what I showed you earlier, there's sharing of antigen across these different DC subsets. But in the skin, if you look at migratory cells that are bringing antigen in, there's lots of antigen present in the migratory cells, but there is essentially a lack of antigen sharing with the resident DCs. And so my question then was, what is kind of the gatekeeper for this antigen passing phenotype? With my first expectation being that likely inflammation within the periphery could be licensing DCs to pass as they enter into the lymph node. And so to get at that, here I just did a experiment that induces general inflammation with low-grade irradiation. So if you give 1 gray irradiation to these steady state mice, what you begin to see is an increase in the presence of ZsGreen within these resident DC subsets, suggesting that indeed peripheral inflammation can induce antigen passing. This has been also, I've also done this with application of DBP, so Horbol ester, onto the skin of the mice locally, and you can again see increases in this antigen sharing with the resident populations. So potentially what you have here is an on-off switch of antigen sharing with resident DCs, and our lab is currently looking at what exactly then the role of antigen within these DC subsets could be for dictating immune responses like peripheral tolerance. So in that data I showed you in the published paper, we concluded that CD8 alpha DCs are resulting in a suboptimal prime. Well, our hypothesis then is that in the case of these resident DCs, there's a potential for these resident CD8 alphas suboptimal priming event to be contributing to the maintenance of peripheral self-tolerance. So in the context of inflammation, the antigen passes to these tolerogenic priming DCs and may influence or at least reinforce peripheral self-tolerance in the context of tissue inflammation. And those are ongoing studies that we're currently diving into. So looking at self-antigen and tumor antigen in the context of peripheral inflammation is an area that my independent group is very interested in expanding upon. And the reason we are so interested in this is kind of centers around a long-held issue that dendritic cells have to decipher, that being how do they decide what and when to present antigen to T cells. What I'm referring to is that in the context of an individual DC, these DCs have very complicated decisions that they have to make in a context in vivo that can be very complicated, meaning that an immature DC, when it takes up antigen, in this diagram here, it's depicted as if there is only one activating event and only one type of antigen at a time, but that's not really the case. So here, this individual DC takes up antigen in a danger signal environment, and this stimulates activation of T cells, or the DC takes up antigen in steady state and then it tolerizes T cells. But in reality, an individual DC in a environment can take up both self and pathogen or self and tumor antigen, and in an environment of inflammation, so it has received all of the appropriate signals to result in activation, but it has to then find a way to decide between presenting self and presenting tumor, or presenting pathogen and presenting commensal. And so in this complicated way, a DC is always having to make these decisions that are crucial for dictating either tolerance or activation. And so my hypothesis is that this means of internalizing antigen, compartmentalizing antigen, may in fact allow a level of additional control for the dendritic cell by allowing it to shuttle antigen into unique compartments that may or may not have access to the antigen presentation machinery. And so my basis for saying that really comes from the literature, in that back in the early 2000s, Ruslan Menzitov's group, when Julie Blander was a trainee there, published papers in Science, and then a few years later in Nature, explaining this ability of a DC to take up both apoptotic self and pathogen, and then only engage in presentation of pathogen-associated antigens while restricting presentation of self-associated antigens. And the way this is all mediated is through an additional signal from the actual antigen internalization itself, meaning the DC's required TLR signaling to be activated within the phagosome that was internalizing the pathogen-associated antigen in order for that antigen to have access to antigen presentation pathways. The apoptotic self cell did not have TLR signaling in that phagosome, and so it was not allowed to access the antigen presentation pathway, and so you did not get responses to self. This work, it was really elegant and beautifully done, and since then, it presents a lot of questions about how this mechanism may be engaged in vivo, as well as engaged in the context of different types of antigen. So some important questions that remain are to look at how this deciphering of different types of antigen uptake influences presentation in vivo. They were looking at apoptotic self cells, but what about apoptotic tumor cells? Can the DC distinguish the difference between self and tumor? And how does this translate to cross-presentation? All of the work was done in MHC class 2 restricted antigens. So what does this mean for CD8 positive T cell stimulation? And then looking further into how all of this kind of sorting is mediated, is there potential for specific receptors that are engaged in different contexts to control the availability of these antigens to access cross-presentation? And so I have developed a couple of different models that will allow me to answer these questions. One I introduced to you earlier in the talk using B16 ZsGreen labeled tumors. We can sort the DCs out. You can find them, the antigen encapsulated with membrane. The other being the K14 ZsGreen self antigen mouse. We can again sort DCs out, find ZsGreen internalized. There's plenty of uptake in the skin similar to the tumor. But we have other ways of looking at antigen that could be dangerous to the host versus antigen that is considered safe for the host. And that is by using different types of bacterial antigen. So we have designed, in collaboration with Tiffany Shard-Smith's group at UCSF and a postdoc in her lab, John Leach, they have shared some of their bacterial strains with us that express ZsGreen or another fluorescent protein and allow us to look at how bacterial antigen is taken up by DCs and compartmentalized. And so here you can see a Staph aureus ZsGreen, which is a pathogenic bacteria that we can colonize on the backs of mice. And then we have, on the other hand, we have a commensal bacteria, so Staph epidermidis, that also expresses ZsGreen or another fluorescent protein that we can use for tracking studies as well. And so here we have this, these two different systems that allow us to, in two different ways, address dangerous antigen, so tumor and pathogen, versus safe antigen, as in self-antigen and commensal. And so just to get into some of the data that we have so far that I think is really interesting is that if you take a Staph epidermidis, so your commensal bacteria, colonize the backs of self-antigen, so safe antigen mice, and then sort out DCs and look for how the DC is handling the antigen, you can see that when you find ZsGreen, that's where you find the bug antigen. So self-antigen, bug antigen are co-housed. So nearly 95% of the DCs that have M-cherry signal, that M-cherry signal, 95% of it is co-localized with self, suggesting that the internalization and holding of this antigen is together inside an individual DC. What's interesting is that that doesn't seem to be the case when you look at two different types of antigen in the case of danger and safe. So if we colonize the backs of these mice with a Staph aureus M-cherry, so a pathogen, sort out the DCs and look at how the DCs are handling the antigen, we see that the DCs are actually very distinctly segregating pathogen-derived antigen versus self-derived safe antigen. So here are the images from those sorted DCs, the self-derived green, the bug-derived in red, and you can see that they are essentially exclusionary. This is just the same kind of experiment except with lattice light sheet to show with greater granularity, and you can see again, these DCs are holding the antigen separately. How much of that is influenced by the virulence of the bacteria? We were curious to know if we made the pathogen more commensal-like, would it then be housed like a safe antigen, like a commensal? And so John from the Shard-Smith group made mutant Staph aureus mice that had the AGR locus deleted. So this made the Staph aureus much less virulent. We could colonize the backs of our K14 ZsGreen mice, look for our cherry bug signal, and found that indeed there was some restoration of the housing of antigen back to a co-housing with safe self-antigen. So here's that what you see with Staph epi safe and commensal, where most of it's co-housed, Staph aureus danger, most of it's separate. And then with the AGR knockout, you can see that you shift back to the majority of the antigen being co-housed with self. So while that's just kind of a taste of the type of data that we are generating and the questions that we're interested in investigating, the conclusions from this work, there are obviously more questions than answers at this point, as probably is the case with many new labs. But what I think we can say is that DCs are able to keep antigens segregated in a pattern that's consistent with safe and danger reservoirs. And so we're very curious to move this into looking at comparisons between self and tumor, given that tumor has both characteristic identity of self, but also is in a context of danger. So what pattern is being assumed by tumor antigen? And so we're interested in giving tumors to our self-reporter mice and seeing where the antigen resides. This brings up interesting questions about early and late lesions. So is there a switch early on in pre-neoplasia where, say, early in tumors, antigen is being handled as self, and then at some point it switches over to a danger signal? How the kinetics of that would be working or is something of great interest to us. And then of course we want to know if this dictates differences in T cell activation versus tolerance. So is the identity of the encapsulation of these antigens on an individual DC level influencing access to the cross-presentation pathways? And so with that, I'd like to acknowledge my lab. We're small and new, so Kartik is a postdoc and Cassidy is a fantastic technician in the group. We have an ongoing collaboration with Tiffany Shard-Smith's lab in all of the microbiology, pathogen commensal work. And then, of course, a big thank you to Max, who has been a very supportive mentor in my transition over into my independent group. Ed Roberts is the postdoc that did a lot of the work on that cancer cell paper with me. And Kai was another postdoc who was integral to some of the imaging analysis. And then, of course, we thank our funders. So again, thank you for the opportunity to present in this forum. And I'm looking forward to hearing the rest of today's talks.